Ever wondered how you inherit characteristics from your parents? It's all in the DNA replication process. DNA, deoxyribonucleic acid, a double helix molecule, is the blueprint for life. It's the carrier of genetic information that's passed from generation to generation. This genetic language in our cells is what makes each of us unique. But how does this information get passed on? That's where DNA replication comes in. This intricate process is a marvel of nature, a microscopic dance that ensures each new cell gets a complete set of DNA molecules. It's a process so precise, so well coordinated, that it's hard to imagine it happens billions of times every day in our bodies. In the following scenes, we'll be peeling back the layers of this process, revealing the magic behind the replication of life's blueprint. So buckle up as we dive deep into the microscopic world of DNA replication. To understand DNA replication, first we need to grasp what DNA is made of. Our DNA, or deoxyribonucleic acid, is the blueprint that guides all biological processes in our bodies. Imagine it as a long, twisted ladder, a shape we refer to as a double helix. Each rung of this ladder is made up of something called a base pair. But let's take a step back. The DNA ladder, or the double helix, is composed of repeating units known as nucleotides. Each nucleotide is a combination of a sugar molecule, a phosphate group, and a nitrogenous base. The sugar and phosphate form the backbone of the DNA strand, providing structure and stability. But the real magic happens with the nitrogenous bases. There are 4 types of these bases in DNA: adenine, thymine, guanine, and cytosine. Remember our ladder analogy? These bases are the rungs. Adenine always pairs with thymine, and guanine always pairs with cytosine. It's this specific pairing, known as complementary base pairing, that forms the rungs of the DNA ladder. These base pairs are the language of life, spelling out the instructions for every protein your body needs to function. The order in which these bases are arranged determines the genetic code. Think of it as the world's most complex instruction manual written in a 4-letter language. These bases aren't just sitting there though, they're held together by something called hydrogen bonds. These bonds are strong enough to keep the DNA intact, but weak enough to be broken when the DNA needs to replicate. And that's crucial for life to continue. So, DNA is a double helix made up of nucleotides which consist of a sugar, a phosphate, and one of 4 bases: adenine, thymine, guanine, or cytosine. These bases pair up to form the rungs of the ladder, and their specific sequence forms the genetic code that guides all life. Now that we understand the structure of DNA, we can delve into how it duplicates itself. The magic begins in a specific region of the DNA called the origin of replication. This is where our story of replication really takes off. DNA, this miraculous blueprint of life, does not replicate all at once. It starts from specific points known as the origins of replication, where the double helix structure of DNA begins to unwind. Imagine the DNA as a twisted ladder. At the origin of replication, this ladder starts to separate, forming what we call a replication bubble. This bubble is the playground where the replication process occurs. It's a bit like unzipping a zipper, where the 2 strands of the DNA helix start to separate. Now you might be wondering who or what does this unzipping. Meet the enzyme called helicase. Helicase is like a molecular motor. It breaks the hydrogen bonds between the DNA strands, causing them to separate. As helicase moves along the DNA, it continues to unzip the strands, allowing them to serve as templates for the synthesis of new DNA strands. But there's a catch. DNA doesn't like to be unwound. It creates tension in the unseparated part of the helix. Enter topoisomerase, another crucial enzyme in our replication story. Topoisomerase works ahead of the helicase, cutting one or both DNA strands, untwisting the helix, and preventing supercoiling. Think of topoisomerase as a stress reliever, helping the DNA cope with the tension of being unwound. So what we have now is a replication bubble with helicase unzipping the DNA and topoisomerase relieving the tension. This replication bubble will continue to grow as more of the DNA is unwound, preparing the stage for the new DNA strands to be synthesized. It's a beautifully coordinated process, displaying the incredible precision of molecular biology. With the DNA unwound and ready, we can move on to the next phase. Now we're at the heart of the process where the DNA starts to duplicate. So what's happening here? Well, the main player is an enzyme known as DNA polymerase. This little molecular machine has a big job. It's responsible for adding new nucleotides, the building blocks of DNA, to the growing strands. Here's how it works. DNA polymerase moves along the open DNA strand, reading the sequence of nucleotides and adding complementary ones to the new strand. It's a bit like putting together a puzzle where each piece must match its partner perfectly. But there's a twist. DNA polymerase can only add new nucleotides in one direction, from the 5' to the 3' of the new strand. This leads to an interesting situation. On one side, the leading strand, DNA polymerase can work continuously, zipping along and adding nucleotides as it goes. It's a smooth, efficient process, like a well-oiled machine. On the other side, however, things are a bit more complicated. This is the lagging strand, and because of the direction that DNA polymerase works, it can't replicate continuously like the leading strand. Instead, it has to replicate in small chunks, moving backward from the replication fork, the point where the DNA is being unwound. These little chunks are called Okazaki fragments, named after the scientists who discovered them. Each fragment is started with a primer, a short piece of RNA, and then DNA polymerase fills in the rest. Once a fragment is complete, another enzyme, DNA ligase, comes along and glues the fragments together, forming a continuous strand. So the process of elongation, this dance of enzymes and nucleotides, is a bit of a two-step. It's a rhythm of building and binding, of reading and replicating that allows the DNA to duplicate itself perfectly, every time. With the new DNA strands forming, we're nearing the end of the replication process. Just as every story has an ending, so does the process of DNA replication. The final stage, known as termination, is just as crucial as the rest. After the new DNA strands have been synthesized, they must be checked for accuracy. This is where the proofreading phase comes into play. Enzymes responsible for DNA synthesis also have the ability to proofread the newly formed DNA. If they detect an error, they can remove the incorrect base and replace it with the correct one. This process is incredibly accurate, with only about one mistake happening for every 1 billion bases that are synthesized. The termination stage also involves the removal of the RNA primers that were used to initiate DNA synthesis, and filling in these gaps with the correct DNA bases. Once all these steps are completed, the DNA strands fully separate, leaving behind two identical DNA molecules. And there you have it, two identical DNA molecules, ready to pass on genetic information. So why does all this matter? Let's take a step back and look at the big picture. DNA replication is not just a scientific marvel, it's the very fabric that weaves life itself. Each time a cell divides, it needs an exact copy of DNA to function correctly, making DNA replication a fundamental process for growth and development. From the tallest trees reaching for the sky to the tiniest microorganisms invisible to the naked eye, every living being relies on this process to grow, repair damaged tissues, and reproduce. Now you might think, doesn't copying lead to errors? But here's where the beauty of DNA replication shines. The process is incredibly accurate, with numerous built-in checks and balances. So, while errors can occur, they're remarkably rare. It's like having an expert proofreader on hand, ensuring the story of life is told correctly every time. DNA replication isn't just about growth and repair though, it's also central to inheritance. Every trait that makes us unique, from the color of our eyes to our knack for music or math, is encoded in our DNA. When cells divide, they pass these traits to their offspring, essentially handing down the blueprint of life from one generation to the next. But here's where things get a little tricky. There's a common misconception that DNA replication creates an entirely new strand of DNA. In reality, it's semi-conservative. This means that each new DNA molecule contains one original strand and one new strand. Think of it as keeping a piece of the past, while creating a part of the future. So the next time you look in the mirror, remember that every cell in your body carries a copy of your DNA, replicated with astonishing precision. And it's this process that makes you who you are, allowing you to grow, heal, and pass traits to future generations. Remember, every characteristic that defines you, from your hair color to your height, begins with this incredible microscopic process.